Hello there, ninth graders. This is Teacher Krishna, and welcome to our biology class for week number 19. Just like every week, thank you a lot for coming to today's online lesson and, of course, for completing all of the assignments that you have had so far. Keep it up, guys. So before we start, just like every week, log into Agilix, enter to the Google Classroom, download the Word document, and save it inside your biology folder as it is specified right below. Remember that it's extremely important that you keep track of the most important things from the presentation and video by taking notes on your Word document while you watch the video, right? It's very important to start with our ending mind, and today's topic is 3.05, and the ending mind is the following. Students will be able to retell historical facts about the discovery of DNA replication, as well as generalities on the DNA structure. Alright, so we're going to jump in directly to some historical information and some generalities about DNA replication. So, first of all, we are going to analyze the Morse code, which is a new language that transmits information with a different code. So, basically what Morse code does is that it's going to transfer certain messages, but with the support of a different code, such as, let's suppose that you use a flashlight. So by turning on and turning off the flashlight, you can transmit a message to someone that actually knows how to understand the Morse code. So something very similar happens to DNA. So DNA also has its own code and its own way in how it is going to transmit information and messages, which is what we are going to be studying today. So let's go with some historical facts about the discovery of DNA, genes, and everything. In 1865, Gregor Mendel demonstrates patterns from parents to offspring. So what Gregor Mendel did, as you might remember, was that he was experimenting with some peas, and through his investigation he was able to identify some patterns that peas will have from the first generations or the original generations to other offspring. In 1903, a student discovered chromosomes. Remember that chromosomes are a combination of DNA. In 1911, Thomas Hunt Morgan discovered a genetical pattern in a fruit fly's chromosome. So this was a huge discovery because he started noticing already the pattern that it was followed in some chromosomes. In 1928, Frederick Griffith discovered DNA in bacteria. This was something amazing because before him, no one discovered this kind of protein because and what happened was that everyone knew that organisms will surpass genetical material from one to another, but they didn't know exactly how. But Frederick actually helped a lot in the discovering of the particle that we will know now as DNA. In 1949, Erwin Chargaff found that the amount of nitrogen bases will be equal in similar portions of DNA. What happens is that DNA is conformed by several nitrogen bases. And especially they have 4 nitrogen bases, which are adenine, thymine, cytosine, and guanine. Those are the 4 paired bases that are going to conform DNA. So what was interesting was that Erwin Chargaff was analyzing the amount of nitrogen bases in animal DNA and in plants DNA. But surprisingly, he found that there wasn't really a huge difference according to the amount of particles that he could find right there. There was no real difference between how many particles of adenine, how many particles of thymine he will find in one or another. So that truly called the attention of a scientist because no one knew how to explain that. In 1952, Rosalind Franklin discovered the double helix with X-rays. This was a huge discovery as well because scientists did not really know the way DNA was structured. But after Rosalind Franklin exposed some portions of DNA to X-rays, he started getting some kind of blurry pictures of what it will represent. But then she discovered that DNA will look like an X or with a double helix. And then in 1953, James Watson and Francis Crick created the first 3D DNA model of DNA. That was something amazing because after they recreated this 3D model of DNA, a lot of scientists could actually identify the way DNA is structured. And lastly, one very recent discovery was in 2003 by Craig Venter and Francis Collins. This was something astonishing. So after 13 years, they were able to map the 3 billion chemical paired bases of the human DNA. That is something amazing. Imagine they spent 13 years just to identify and actually create a map of all of the billion chemicals that the DNA has. Of course, in the human DNA. Okay, so let's continue. So this is what the model of that the scientists presented before will look like. But this is something very simplistic. So if we analyze it at an atomic level, we actually will get something a little bit different that I will show you later on. So now we have some important concepts. We have nucleotides, so macromolecules composed of deoxyribose, a phosphate group, and a nitrogenous base. They are together by covalent bonds. So this is the first concept that we need to analyze, and it's something very important because it's part of the DNA structure. We also have nitrogenous base, which are bases that contain nitrogen. And just like I told you, these bases are the ones that conform DNA, mostly. And these are like the tubes that you can see in the picture right there that are in the center of the double helix. Those tubes are the paired bases. In total, we are going to find 4 bases in the DNA, which are adenine, which stands for A. We have thymine, which stands for T. Cytosine, that stands for C. And thymine. Oh, it should be guanine. I'm sorry. There should be a letter G in there. Guanine in the last one. So adenine is always going to pair with T, which is thymine, and cytosine is always going to pair up with guanine, which is a letter G. They work as the alphabet. So we only have 4 paired bases. And just like in our vocabulary, where we have 26 or 27 letters, depending on what language we're talking about, we can recombine those letters to create a huge amount of words. And we can communicate several messages. The same thing happens with the paired bases. So you have only 4, but once you recombine them, you can create, trust me, almost infinite recombinations. And now we have hydrogen bonds. They are weaker than covalent bonds, but they are unique nitrogenous bases. So separating the strands of DNA is essential for DNA replication. So when adenine pairs up with thymine, what happens is that they are going to get together by a hydrogen bond, okay? The same thing happens when cytosine is going to be pairing up with guanine. They are going to be getting together with a hydrogen bond. That is not really that strong because DNA needs to separate to actually replicate later on. Here you have an example of what the replication of DNA looks like. We have the original DNA, so we have our 2 strands of DNA, and also we have several paired bases, and as you can notice, adenine is always going to pair up with thymine, so A is always going to be with T. And cytosine is always going to be with guanine, so C is always going to be with G. Okay, so now, as you can see in the next step, original DNA strands prayed apart. And we have one green part at the top and one green part at the bottom. So how is it going to replicate? So some extra paired bases are going to come and they are going to attach to the original ones. But remember, they cannot attach in a different way. Thymine is always going to pair up with adenine. And cytosine is always going to pair up with guanine. It is not possible to get any other combination. Okay? So that is why two identical DNA molecules have new and have from original form at the end. Alright, let's continue. Now we're going to analyze a very important enzyme, that is a polymerase. So paired bases allow DNA to recreate exact copies of each other. This is something extremely important because remember that DNA has two strands. So whenever it separates, it cannot get like a different copy or like a different combination, because the bases already have one that they have to pair with. That is why it is possible for DNA to create exact copies. The two strands of DNA are defined as complementary. They are complementary because one cannot exist without the other. And basically, they are pairing up extremely specifically, one paired base with another paired base. And before a cell divides, it replicates its DNA. Remember that we already studied the process of mitosis? So before the division of the cell starts, and even before the whole process starts, the DNA is replicated in the nucleus. So at the end, the daughter cells are going to get the exact amount of genetic information on both sides. Enzymes break down the hydrogen bases. So remember that the paired bases have hydrogen bonds right in the middle. So enzymes are going to break through those connections from hydrogen, so then DNA can replicate. And DNA polymerase is an extremely important enzyme because it joins nitrogenous bases from the complementary strand. And it double checks that the new DNA is perfectly copied. Okay? So DNA polymerase is in charge of like pairing up all of the sequence. Let's say that you have a letter A. So DNA polymerase is going to pair it up with a letter T. If DNA polymerase sees C, it is going to pair it up with a letter G. So it is an extremely important enzyme. And when it finishes copying all of the, or most likely when it finishes attaching the new nitrogen bases one to another, it double checks that it is a perfect copy of DNA. Okay, so now please complete the exercise from page number 3 about paired bases. So you can go right there to page number 3. Once you are done with the exercise, you can come back to the video so we can continue with some other important information. Alright, so now we are going to analyze something else, which is DNA and RNA. What is the difference? DNA is the one that we can see on the right. You can see that it has a double helix or a double strand. But RNA only has one strand, which is the one on the left. And let's analyze something important about it. So RNA copies information from the DNA and takes it out of the nucleus. So DNA already has some important information about how much food the cell needs or what certain functions it needs to take place in the cell. So RNA is going to copy the information and then it is going to go out to the cytoplasm of the cell and have some other functions in there that we're going to see. RNA only has one strand. Just like I mentioned before, it contains ribose instead of deoxyribose. That is one of the differences that you can see in between RNA and DNA. DNA contains deoxyribose, but RNA contains ribose. Okay. And so we have the RNA contains uracil instead of thymine. We have to remember that in the DNA we have four components, which will be adenine, thymine, cytosine, and guanine. But in RNA, what is going to happen is that thymine is going to be substituted by uracil. So we're going to have in RNA adenine, uracil, cytosine, and guanine. And whenever these are pairing up, uracil is going to pair up with adenine, because remember that it is substituting thymine. Okay, let's continue. So we have three different types of RNA. We have messenger RNA, which copies the information from the DNA, just like I mentioned before. We also have transfer RNA. It reads the codes from messenger RNA and attached amino acids to create a protein. So what the transfer RNA is basically going to do is that it is going to read the codification from the messenger RNA. So it is going to be analyzing like groups of three bases from the messenger RNA. And based on the combination of the three nitrogenous bases, it is going to attach a certain amino acid in order to create a protein. But they still need to work together with a ribosomal RNA, which are organelles that build up proteins. So the three of them are going to work together in order to create new proteins. All right, so now let's go with your evaluation. Read and summarize pages 1 to 5 of topic 3.05. Especially focus on page number 5. I included most of the information from pages 1 to 4, but you need to read one last page to add more information to your summary. Then add images and some extra decoration to your summary. Upload your summary to Google Classroom. What is the rubric that I'm going to use to evaluate your summary today? The order will be 1 point, grammar 1 point, images 1 point, meaningful information 1 point, and lastly the format of the summary will be 1 last point. So it's been a pleasure to have you here with me today, guys. I will be seeing you in a couple of minutes during the Zoom session. Goodbye and take care.